हम्म, तो समझो भाई।
Good evening, everyone. I will just wait for another 5 minutes more, and allow others to come. Okay, and then we will start with the class. So I guess I'm audible. So good evening to one and all present here, right? So let me start by introducing myself to you all. So I am Srijan Sen. I am a PhD scholar from IIT Kharagpur in the Department of Bioscience and Biotechnology, and I will be taking your live session on the course Introduction to Complex Biological Systems, which is being taught by Professor Shomo De and Professor Dibyendu Shamanto of IIT Kharagpur. Okay, so I hope you will enjoy. You are enjoying the course in the starting few weeks, as well as you will be enjoying the live session. And I want the live sessions to be more interactive, so feel free to ask me any questions about this course and things. Okay, so basically, if I give you a brief introduction to this course, or an idea about how this course will go. So usually how I do is, I first of all try to give a brief idea about every week lecture as it goes week wise. So for week one, I would be picking up a few topics which are very very important for you guys to know, as well as there are some interesting stuffs which might not be there in the lecture series. So therefore I put up some few of that, and just because this is more of a interactive course, therefore I tried to design it in a manner that there are a few questions in the form of sometimes maybe MCQ, sometimes maybe MSQ, sometimes more of an assertive reason type of questions that are definitely going to help you in most of these competitive exams, not only this NPTEL exam but also in the different competitive exams as well. So if I give you or if I start telling about this course, that is the Introduction to Complex Biological Systems, though it the name itself has complex, yet it's not that complex. It's not designed in that complex manner. So this course is basically designed for people who come from a non-biology background as well as a biology background, and do not have a much idea about the different topics of biology, and are interested to know more about it. So it's designed in a much more simplified version. So most of the people coming from non-biology background, or backgrounds where people want to shift to the different zones or the different sections of biology, it's a very important or a very helpful course for them. So we start from the very basic nucleic acids, the genetic material, and we go to the we will moving on to the different types of uh, like you have the translation, replication, transcription. So there are different topics which you have heard of when you were in your class 11, 12, and so this is a more depth of that study in this course, and this is designed in such a manner like it does not get more complicated, rather it will be more simpler as we go on week by week and lecture by lecture. So I am not going into in which week what you will be taught, but what I can tell you is that most all the important sections that are required for a person or a scientist, rather a biologist, to know everything will be covered in this particular course. Okay, right. So yeah, I think okay. So when I go into the discussion, or the where I show my slides and start the class, you can ask me questions anytime. It's not that you have to wait till the end before I reach the end and then you ask the questions. You can stop me, and then you can ask questions, not a big deal. All right. And then so I will first cover a few of the important or interesting topics, and then I will be moving on to the question and answers in the end. That will be more of an interactive, where you guys will be, I will be giving up the questions and you guys will be answering it to me, right? Okay. Anyone has any questions about this particular till the part what I have said? Okay. So I think the brief introduction about the course is clear to most of you. Also, what you can do, like even if I miss sometimes, I might miss a few questions. People are trying to say the questions, and I might miss it while I'm teaching. Then you can also put it up in the chat box if that's more comfortable with you. So there's nothing to feel bad about. It's a simple question, or it's a very weird question. Nothing is weird over here. So we totally understand, though I don't know from the backgrounds from which you all are coming. Yet I believe that there are people from different backgrounds as well. So it's absolutely okay to put up any question. Okay, so feel free to ask questions, because this is an interactive session and I want you all to be interactive. Okay, right. So I'm sharing my slides with you all. Okay, so is my slide visible to you all? Kindly confirm. Okay, cool. So basically, in today's class, we would be talking about nucleic acids. That is the basic and the hard drive for the information of life. So this is the topic that we are going to discuss in today's class. So how do we start? So the discovery of nucleic acids, so there is a huge history of how the nucleic acids were discovered, but the basic idea behind the discovery of nucleic acid was the search for the genetic material. So people, or rather scientists, researchers, were hunting for the material or the substance that is holding all the information. And when they went on to find, like, you have to find out where is all the informations, or where all the informations are being stored, you have to search for the substance that keeps or stores all the informations, right? So in the search for the genetic material, there are some substances that must be responsible, because there were passage of traits, properties from the parents to the offspring. So for a substance to do this, so there are certain properties that the substance must maintain. So like, it should be stable enough to store the information for a longer period, and it is being carried from decades, from generation after generation, for long term. So it should be able to do that. It should be able to replicate accurately, such that the genes are being transferred from one organism to the other. So the relevance of these things is totally based upon what is the thing that is being maintained over the generation after generation, or where is it being stored, and how even after evolution. Okay, so there is an evolution that is also going on every, like 5-10 years. There is evolution. Now you most of you know there is a Gen Z, there is a Generation Z, Generation Alpha. So these are quite terminologies used nowadays. So there is an evolution that is happening constantly, right? So it should be stable enough to hold the information, replicate properly, as well as it should be able to uh, understand the evolution and pass on the trait. So, what constituent of the cell carries the genetic information? That was the next question. So, from the genetic information, the term gene comes. That's a high stability requirement capable of sudden change to mutant forms to provide the basis of evolution. Now, if there are few changes within the genes, now what are these genes? So, then the genetic information. So, these are quite complicated terms. Let us not consider those things as of now. If you have an idea, so that's fine. But these genes are basically the material where the information is being stored. Fine. So there are some, like you have some computer languages. If you know, you have heard of Python, C++. So there are some computational terminologies that are being used. So there are some must be some portions within the genes or some constituents within the genes that holds this information. Okay. So from the idea about what the cell is comprised of, as well as what is the genetic information, where is it stored, how do we get it, the concept of genes come. Then there was an idea about proteins, DNA, and RNA. So these were different studies. Why is it important to know? Like, it should be understood how and where the information is being actually stored. So scientists were trying to figure these things out, and then they found out proteins, where they found strong evidence initially in favor of proteins by Garrod and Beadle and Tatum theories of genes controlling the enzyme synthesis. So they thought that okay, there might be some proteins within the cell that are playing an important role. Next, what they tried to do is now then they came across nucleic acids. Now nucleic acids are of two types, the DNA and the RNA. So DNA, so some people said that okay, DNA is probably the portion within the cell that carries the genetic information. Okay, so that they found out using the Griffiths experiments and later on by Avery's experiments to prove that DNA has a genetic material in the cells. And then comes the RNA. So people said that okay, so RNA is the genetic, is the thing that is storing the information. So different scientists have found out different proofs from which they can tell that the genetic information is being stored in proteins, in DNA, as well as in RNA. So is this hypothesis true, or are these scientists somewhere doing something wrong? To be it sounds quite rude, yet what was the actual issue with this? Because some scientists found that it is being stored in protein, some people say no, it's stored in the DNA, some are telling in the RNA. Why is it so? So to answer this, there is the central, there comes the concept of the central dogma. Okay, where you have a starting material, that's the DNA. Okay, it gets transcribed to an RNA and then translated to the protein. We have somewhere heard about the central dogma. So it is not that the genetic information is only stored in the DNA. So most of you have heard about that egg and hen story, or the controversy. So what came first? Is it the egg or the hen? So biologists have a great controversy about what came first. Is it proteins, DNA, or RNA? Which one came first? So there is always a controversy, because why do I say so? It is because you see, in the different stages of, say, a DNA replication, or you say a transcription, even a translation, there is always a role of some proteins or the other, be it enzymes, which are also proteins. Then you have the RNA, the transcription process, or even the DNA replication process. All of there are proteins, but the in some stages, like the DNA replication, DNA is the starting material, so there has to be some genetic, there has to be some DNA, or even in translation there has to be some RNA, be it a messenger RNA or some RNA, some coding RNA strands through which it has to translate. So what came first? So what is the starting material that was there? Okay. So there are a lot of controversies going over there. We need not worry about it. This, so it means that there is an information that is present in some form. It is transcribed to the other form and translated to the final you can say the final structure or the final form of it. Correct? If that is true, so the different scientists, none of them did anything wrong. They were studying the different parts of the cell. Some were studying the DNA, some were studying the RNA, and some were doing studies on the protein. And they found that each of these components of the cell can hold the genetic information, but in some different forms. So some people, what they do, they write in so when someone is maybe sad, what they do, this someone writes in a diary, someone sings a song, someone has a different way of expression. Okay, so these DNA, RNA, and proteins, they have different ways of expressing or storing of the genetic information, right? And this is very important to know before we go into the depth of DNA, that's the genetic material in the living cells. Okay, so to start with, there is a history where most of you might have studied about the Frederick Griffith's 1928 experiment. So he did experiments with the Streptococcus pneumonia bacteria in mice, which showed that something passed from the dead bacteria into the living ones, allowing them to change their cell surface. And he called this agent the transforming principle, but he had no idea how it worked. It was long back about this Griffith's experiment. So what he basically did was he took the living or the virulent bacterial strain, injected it in the mice, and what happens? So you have a virulent strain of bacteria, you inject it in the mice, it dies, right? Then you take a living rough colony or an avirulent strain, so the strain that's not virulent, and you inject in the mice, so the mice lives. That's quite understood. Okay, so you have a virulent strain, you inject it into someone, the mice dies. You have an avirulent strain, you inject it in the mice lives. That's fine. But what happens next? The next two experiments are very, very interesting. So how is it so? So it takes so what Griffith did was he took the virulent strain and he heated it. So when you're heating a virulent strain, what happens? It kills it. So he first heated it and then he injected it in the mice. The mice lives. You can understand that. After heat killing the virulent strain, the effective or the efficacy of that strain basically decreases and the mice lives. But next what happened is that he took the living avirulent strain. Okay, So he takes an avirulent strain and he takes the heat killed virulent strain. So now, if you can understand that both of these strains are avirulent in nature, okay, because in both the cases the mice lived, right, when they were transformed or injected individually. Now what he did, he mixed them and he injected it in the mice. The mice died, and what he observed was living virulent strain inside that mice when the tissue was analyzed. So now the question was, what was the pushing condition or the driving force that occurred, or what was the exchange that occurred between the heat killed virulent strain and the avirulent strain that not only led to the death of that mice but also activated the heat killed virulent strain within that particular mice. So this is another experiment of Griff. So this is a similar experiment, but he did it. So you can see over here. So there is a virulent strain of bacteria that's present over there, and then you have the heat killed avirulent strain. So you have a virulent strain and a virulent strain. So this is a, you killed it and then you injected it. So now what happens, you see that there is a portion of the gene. So we call, so this marked green portion you say, or you say the yellow portion, we basically call them as inserts or segments of the particular gene which can undergo recombination. So what happened, this heat killed, right, so this heat killed genetic material was present freely. What happened, a virulent and a virulent strain have a very similar kind of a genetic, you can say the genetic arrangement, or genetic, maybe a mutation here and there is possible, but more or less the genetic signature is quite similar. So what happened, this particular segment of the gene was substituted with the open fragment, the fragment that was heat killed. So it was present freely, it got exchanged with this particular portion that was avirulent, and now that it has found it has found a different body, there was recombination, there was cell division, and the strain, although being heat killed, so heat killing does not lose the information, it loses the structure, it breaks apart, but the ultimate material is stored, it finds a body. So it's more similar like a person dying, we say that the soul never dies, so it's similar to that, the message is never lost, the genetic information is never lost. So that is why DNA is the material that keeps or stores the genetic material, because it's never lost. When it finds another body, it finds another, we call these stuffs as plasmids, it finds another plasmid, it's again active. And from here he said that DNA was the genetic, was the basic unit of storing the material. Next up is the Hershey-Chase bacteriophage experiment. So this is another evidence where DNA was shown as a genetic material, and this was done in the 1953 by Alfred Hershey and Martha Chase work on E. coli infected with bacteriophage T2. So what he did, in one part of the experiment, the T2 proteins were labeled with ACE35, so it's an isotope of sulfur, it was labeled with that, and the other part was labeled with 32p. Why he did that, we will come to this. So then what they did, then each group of the labeled viruses were mixed separately with the E. coli host. Now, after a short time, the fudge attachment was disrupted with some kind of an instrument like a kitchen blender or things like that, and the location of the label was determined. So what he saw that what he saw was basically he found that outside the infected ACE35 labeled protein was found outside the infected cells, while the 32P labeled DNA was inside the E. coli, indicating that DNA carried the information that was needed for the viral infection. And this uh, provided additional support for the idea that the genetic inheritance occurs within the DNA. So you see the bacteriophage, so it's an unlabeled one, and then it's being labeled with a 32P and a 32S. So it also indicates that the genetic material can be passed on to the E. coli cells, and because of that the genetic material, so what does the genetic material contain? So the genetic material contains a phosphate group, because DNA has a phosphate group, and that phosphate group is 32P labeled, and because of that the DNA that was found inside the E. coli, so bacteriophages are known to infect bacteria, correct? So it transfers the genetic material into the bacteria and then it gulps it, right? So the ACE32 is a part of the bacteriophage and it was found outside the infected cells, while the 32P, that is the important part of the DNA or the genetic material, was found inside the E. coli. Okay, so you see over here, so it's labeled, so the genetic material has been transferred. Okay, so the phage ghosts you see over here are labeled with S35, so the its structure contains that S35. Okay, and the material over here is unlabeled, and from outside you see it's labeled because the covering contains the sulfur group, and this sulfur group is labeled, whereas when you are labeling the genetic material, it's infecting the bacteria, so it's transferring or putting its genetic material into the bacteria. You see, you find labeled phosphorus. So this is the same thing that is defined in a more just manner. So what you need to understand is basically that the protein that defines the structure of the bacteriophage is made up of the sulfur that's labeled or unlabeled, so it's made up of the sulfur, whereas the phosphorus is 32P and that is the composition of the DNA or the genetic material. Now, so when we talk about the composition, the structure of the genetic material, we need to understand it. So nucleic acids are of two types, the DNA and the RNA. So DNA and RNA are polymers composed of monomers called the nucleotides. So what are nucleotides? Nucleotides has three parts. The pentose sugar, that's a 5 carbon. Then you have a nitrogenous base that can be also categorized into 4 types, 5 types. And then you have a phosphate group. When you do not have a phosphate group, that is only the pentose sugar and the nitrogenous base, what do you call it? You call it a nucleoside. So nucleoside is a pentose sugar and a nitrogenous base, and a nucleotide is a nucleoside plus the phosphate group. So the pentose sugar in RNA is ribose, and for DNA it's deoxyribose. So deoxyribonucleic acid and ribonucleic acid. So you know most of it. The only difference is that the 2' position where the RNA has an hydroxyl group, where DNA it's in deoxy, so it does not have that OH group at the 2' position. That's the difference. Okay, so from the structure you can see over here, It does not have that OH group, whereas this has the OH group. So like I was speaking, so the nucleoside is basically the nitrogenous base plus the pentose sugar for the DNA and RNA. Both are applicable over here. Then what happens is that there is a loss of water, and then there is joining of this phosphoric or the phosphate group to this particular 5' carbon and the nitrogenous base to the 1' carbon, and then there is a glycosidic bond formation, phosphoester bond formation, and most of you know that there is a phosphoester bond as well as a glycosidic bond. So the glycosidic bond is between the ribose sugar in the 1' position and the nucleo and the nitrogenous base, whereas there is a phosphoester bond between the 5' carbon of the ribose sugar and the phosphoric acid. So this is a nucleotide because it has a phosphate group, it has a 5 pentose sugar, and this particular nitrogenous base is basically adenine, so we call it DAMP. Okay, now there are two classes of nitrogenous bases that needs to be discussed and remembered as well, where you have the purines, that's a double ring 9-membered structures that includes adenine and guanine. You have the pyrimidines, that's a 1 ring 6-membered structure that includes cytosine, thymine in DNA and uracil in RNA. Okay. Okay. Now, so basically, how do you actually remember it? That is very, very important. So you know purine. Purine is quite. So if you see the number of alphabets in purine and pyrimidine, so purine has less number of alphabets. So it means that it has a bigger structure, whereas pyrimidine has more number of alphabets. So it means that it will have a it's a more simple structure. Okay, so it has 6 carbons, and over here you have how many? 8 carbons, so basically 9 atoms, and over here you have 6 atoms, right? And then under the category of purine you have adenine and guanine. So what are the changes? So in place of this particular, so in place of this 6 hydrogen number 6 attached to carbon 6, that is substituted with an NH2 group or an amino group for an adenine, and for a guanine over here you see that the hydrogen attached to carbon number 6 is replaced with a double bond O or a carboxyl group, and in carbon 2 you have an attached amino group. So that's guanine. So this is about adenine and guanine, part of the purines. For pyrimidines, it's basically so the structure is quite simple. So over here you have cytosine, uracil, and thymine. So uracil is present in RNA and thymine is present in DNA. So cytosine, what you see over here, you have that 4th carbon hydrogen that is substituted with an amino group, and in number 2 position you have an carbonyl group, whereas for uracil there is a carbonyl group. So you see over here all these 3 structures basically has a carbonyl group in position number 2, and for cytosine there is an NH2 group at position 4. For uracil you have another carbonyl group at position 4, and for thymine you have a carbonyl group at position 4, as well as at position 5 there is a methyl group. Okay, so you have to remember these structures. So there are different ways of how people try to memorize these structures. It's absolutely fine how you want to proceed. How I basically did was a smaller alphabets, so a bigger structure, and even less number of secondary structures are there, like adenine and guanine, and pyrimidine has a more number of alphabets, so it has more number of secondary like cytosine, uracil and thymine, and it has a more simpler structure, right? Okay. So the next part is the structure of the nucleotides has these features. So the base is always attached by a covalent bond between the 1' carbon of the pentose sugar and a nitrogen in the base, specifically the 9 nitrogen in purines and the 1 nitrogen in pyrimidines. And the sugar base combination is a nucleoside where when a phosphate is added always to the 5' carbon of the pentose sugar, it becomes a nucleoside phosphate or simply a nucleotide. Very important. And polynucleotides of both DNA and RNA are formed by stable covalent bonds, that is the phosphodiester linkages between the phosphate group on the 5' carbon of one nucleotide and the 3' hydroxyl on another nucleotide, and this creates the backbone of a nucleic acid molecule. And the other thing is that the asymmetry of phosphodiester bonds creates a 3' 5' polarity within the nucleic acid chain. Okay, so now I guess this slide is quite clear to all of you. Okay, and this is the chemical structure of the DNA and the RNA. So mostly how we would say is that DNA, you have the DNA nucleotide, you have the base adenine that is attached. So nucleoside is the sugar plus the base, and then there is a phosphodiester bond over here, right? So there is a phosphodiester bond, so this bond and this bond, and there is a glycosidic the glycosidic bond present in this particular position, right? And for RNA, so the difference between the DNA and RNA is that mostly DNA is double stranded, RNA is single stranded. The stability of a DNA is much more compared to that of an RNA because it does not have that particular 2' OH group which can be reduced or which can be exploited, forming multiple ring structures and stopping the reactivity. So not having that 2' OH group is sometimes quite effective for the long-term storage of material within the DNA more than an RNA. Okay, and the other difference, the structural difference, is that when there is an uracil in place of a thymine, it means it's a RNA, not a DNA. So phosphodiester linkages is basically the repeating sugar phosphate backbone of the polynucleotide chain, and there is a DNA polarity that's defined by the asymmetry of the nucleotides by the way they are joined. So if something goes from 3' to 5', the other chain must go from 5' to 3'. So if one strand goes from 5' to 3', so there is a 5'. What do you have over here? So you have a 5' phosphate group and a 3' OH, right? 5' phosphate, 3' OH. So the other strand, so there are two strands. This is one particular strand, 5' to 3', and the other strand is over here. You can consider 3' to 5'. Okay, so consider from any side. So there is a DNA polarity due to the asymmetry of the nucleotides, correct? Okay. And what does so we know that you have 4 nucleotides that can be present in any way or the other, like DNA or RNA, whatever that is. So it means that for DNA it is, so for DNA you have adenine, guanine, cytosine and thymine, and for RNA it is adenine, guanine, cytosine and uracil. So there are 4 combinations in which they can be present within the for carrying the genetic material. Okay, So like it can be AGCT or AGCU in place of a RNA. Right. Now, what does Shargov's rule say? That in the DNA of each species, the amount of adenine equals to the amount of thymine. Therefore, the percentage of adenine is equal to the percentage of thymine in the DNA, and the amount of guanine is equal to the amount of cytosine. So why is it so? Okay, let us see over here. So adenines can only form hydrogen bonds with thymine, and that also two hydrogen bonds, whereas cytosine can form bonds only with guanine, right, and that also three hydrogen bonds. So mostly we can tell that the number of adenines is equal to the number of thymines. Why? Because DNA, as we generally say, is a double stranded biomolecule. So if it's a double stranded molecule, that means for every adenine present in the strand, so if this is strand 1 and if this is strand 2, so if in the first strand there are, so for every adenine there would be a thymine in strand 2. For every thymine in strand 1 there would be an adenine in strand 2. And similarly for the cytosine and guanine, they complement each other. Just because they complement each other, we call the first strand as the major, or the call it as the template strand. Okay, so we need not remember which one. So from the 5 prime to the 3 prime, we call it the we can call it the forward strand. Okay, so the leading strand, some people call it the lagging strand. Okay, so there are different names to this strand. So we need to know that for every strand there is a complementary strand that is present in the DNA. Okay, because it's a double stranded, and number of adenines is equal to number of thymines, and number of guanines equals to number of cytosine in a DNA. Right. Okay. Another important thing is that there are different DNA segments or different DNA strands that have different melting points, the stability of that particular DNA. What does the stability of the DNA majorly so what does the stability of the DNA indicate, or in other words, what is the melting temperature of the DNA or a nucleic acid? So how can we tell that? So upon heating a particular fragment of a DNA or a particular say material of DNA material, the time when 50% of the DNA has unfolded. Okay, so when 50% of the DNA has unfolded, we call it the melting temperature. But that we all know what is the melting temperature of DNA, that by definition. But actually, how will the melting temperature be different for different segments of DNA? And on what does it depend? It depends on how many cytosines and guanines are actually present. Okay, why is it so? Because if you can see from the structure, there are two hydrogen bonds between an adenine and a thymine, and there are three hydrogen bonds between cytosine and guanine. So the amount of heat that will be required to break three hydrogen bonds should be definitely more than that required for breaking of two hydrogen bonds. So the number of GC pairs, more will be the melting temperature of the DNA, or more will be the stability of the DNA, right? So we just need to keep this thing in mind. Okay. Right. Now let us Okay. So, any questions till here? Anyone has any questions up to this particular portion? So, I guess it was clear to you all. Uh, Anything in the chat box? Yeah, okay. Okay. Uh, Now, before we get into the little bit of DNA replication and all, we first talk about the basic principle, that is base pairing to a template strand. So, since the two strands of DNA are complementary, that means there is one strand from the 5 prime to the 3 prime, and there is another strand from the 3 prime to the 5 prime, or like for every adenine there is a thymine, for every cytosine we have a guanine. So it means that there is a complementary strand of DNA. So each strand acts as a template for building a new strand during replication. DNA replication is important because more than copies, you have to increase the copies of the DNA. So it's a natural process of increasing copies of DNA, because the more the copies, the more the information will be stored for a longer generation, and there will be multiple copies of the information. So the ability to transfer the information will be lot more. Therefore, replication is an important phenomenon. Now, in DNA replication, the parent molecule unwinds and two new daughter strands are built based on the base pairing rules. So, you need to remember that the base pairing rule has to be always followed. That is, what's the base pairing rule? For every adenine, there is a thymine, and for every cytosine, you have a guanine, and that has to be always maintained. So, even if there is a breakage in the strand, there has to be some nucleotides that will come and join over here in the similar manner as the base pairing rule says. So the Watson and Crick's semi-conservative model of replication predicts that when a double helix replicates, each daughter molecule will have one old strand that belongs like the mother strand, and then the one is newly made, or the daughter strand. So there are competing models where the conservative model had the dispersive role. Now, to get started with the DNA replication, so the basic concept is that the base pairing rule has to be followed. To get started, we need to understand, so for a replication to start or a replication to originate, there should be an origin of replication within the DNA. So what is this? So replication begins at special sites called origins of replication, where the two DNA strands are separated, opening up a replication bubble. We know that it's a closed, so mostly the genetic material is a closed structure. So if it opens up at a particular point, right, it does not open up entirely like an open loop. Okay, so there is a portion that first opens up. So most of these are coiled, so they are coiled structures. So when you open up one coil, there is a bubble formation, where there is some kind of stringency in the other parts. So an eukaryotic chromosomes may have hundreds or even thousands of origin of replication. At the end of each replication bubble, there is a replication fork, a Y-shaped region where the new DNA strands are elongating. Okay, so you see over here, there is a bubble formation, and this is a replication fork that is generated. So you see the light colored lines are basically the daughter strands, and this is the mother strand. So it's a simple way of actually portraying it, but it's not that simple. It's mostly remains coiled over here. And then there is the origin of replication. So this is the origin of replication, where the bubble formation happens, and then the it starts complementing each other, and then it grows. Now, first of all, the formation, the origin of replication has to be identified, or the replication process starts with the bubble, and we know that the process of replication is starting or beginning. Now there is an elongation. So elongation is what? So first there is opening up, and then there has to be some kinds of enzymes that will help, that is the DNA polymerase, that will catalyze the elongation of the new DNA at the replication fork. Because it opens up, again it can close, but there has to be some enzyme. So enzymes are basically proteins. So proteins, there are some kind of proteins or enzymes that will, like the DNA polymerase, that will help to catalyze the elongation. So it will add nucleotides to the mother strands and start generating the daughter strand. So the rate of elongation is about 500 nucleotides per second in bacteria and 50 per second in the human cells. So what happens? So there is an anti-parallel elongation that also occurs. Why? So what do I mean by an anti-parallel elongation? So there is always, so you have two strands of DNA. You have a 5' to 3' and a 3' to 5'. But DNA polymerase can only elongate from the 5' to the 3' direction. Okay, that means it will take into consideration the 3' to 5' strand and start elongating from the 5' to the 3' end. But what happens to the other strand that is running from 5' to 3'? So that means there is an anti-parallel elongation that is happening. So that means it will start from the other end, that is the 5' to the 3', and it will start. Because they are complementary, they run anti-parallel to each other. So the daughter strands are anti-parallel. Why? Because they are ultimately complementary to the mother strand. 5' to 3' will generate a 3' to 5', and the 3' to 5' will generate a 5' to 3'. So anti-parallel elongation. But just as I was saying that DNA polymerase can elongate only in one direction, that means if it has to generate something for the 5' to the 3' strand, then it has to generate small fragments, because it cannot generate from the 3' to the 5' end. So the 5' to the 3' will have smaller fragments that will be complementary to the mother strand, and these small fragments that are formed from the mother strand of the 5' to the 3' strand are known as the Okazaki fragments. So along one template strand of DNA, the DNA polymerase can synthesize a complementary strand called the leading strand, that is usually happening for the 3' to the 5' end. Why is it a leading strand? Because there is a continuous elongation that is happening. But for the other strand, that is the lagging strand, the DNA polymerase must work in a direction away from the replication fork. So for that, smaller segments have to be created. That's the Okazaki fragments. And if you're creating smaller fragments one at a time, the process will be less and the elongation speed decreases, due to which it is lagging behind one particular strand, known as the lagging strand. And the lagging strand is synthesized as a series of segments called the Okazaki fragments, which are finally joined by another important enzyme, the DNA ligase. So see 5 prime to 3 prime, there is a leading strand from the 5 prime to the 3 prime direction, considering the replication fork is in this direction, and you have a 3 prime to 5 prime end. So how it generates over here because the replication fork is arranged in this particular direction. So it will start generating over here, then it goes over here, then again over here. Okay, because it cannot generate entirely, and at the end you will see that this particular strand is growing and growing, but this particular strand is somewhere lagging behind because it's a time taking process. Ultimately at the end of the day everything is fine, but it lags behind one particular strand. So this process is done by the DNA polymerase, the elongation, and the Okazaki fragments are joined by the DNA ligase. This is a very important slide where there are some important proteins that assist the DNA replication, which is important for you to know. The DNA polymerase cannot initiate synthesis of a polynucleotide, they can only add nucleotides to the 3' end. The initial nucleotide strand is a short one called the RNA or RNA primer. An enzyme called the primase can start an RNA primer from scratch. Only one primer is needed to synthesize the leading strand, but for the lagging strand, each Okazaki fragment must be primed separately. So DNA polymerase, what it does is it cannot synthesize polynucleotides on its own. So there has to be something like a primer, which is an RNA primer, a single strand small nucleotide sequence about 20 to 25 nucleotides long. So it goes and sits in the mother strand, and from there on it starts elongating, and that happens for a leading strand. But for a lagging strand, every time the primer has to be generated and then it elongates. It cannot elongate for more because these are short fragments. It cannot elongate in this particular direction. Okay, so if a primer maybe, so these two are the primer sequences and it elongates over here, and again it designs a primer for this particular portion and it elongates over here. So then you have then another role is the of DNA ligase, where it joins the Okazaki fragments, and these primers are very important because that's the initiation primer. If it cannot make the, if the primase cannot make the RNA primer, then the replication process won't start, because DNA polymerase cannot make the primer, it can add nucleotides to that particular primer. Then you have helicase that untwists the double helix and separates the template DNA strands at the replication fork. So then you have the SSBPs. Most of you have heard about it. That's a single strand binding protein. It binds to and stabilizes the single stranded DNA until it can be used as a template, because single stranded moieties are mostly weak and are not very stable, so it holds on to that till the entire template formation is done. And finally you have the topoisomerase that corrects the overwinding ahead of the replication forks by breaking, swiveling and rejoining the DNA strands. So like if you take a rubber band and you make a lot of spirals around it, and then from the middle you try to open it, you will see that there are more turns that are happening at the ends. So topoisomerase basically loosens up that tightness, and it loosens up from those ends by sometimes it breaks it, sometimes it untwists it, and it gives it a lot of more comfort. So the primase synthesizes an RNA primer at the 5' ends of the leading strand and the Okazaki fragments. DNA ligase joins the 3' end of the DNA that replaces the primer to the rest of the leading strand, and also joins the lagging strand fragments. Okay. So, yeah. So, what you need to know about replication, most of you have studied about replication even in class 11, 12. But what is more important over here is basically what I have put in over here is the which are the important proteins that play an important role in DNA replication, and which of the proteins comes after what. How do you start the replication? Like, you start from the origin of replication, then there is the formation of this replication bubble. Okay, then comes the primase that has the RNA primer. It sits along the mother strand, and then the DNA polymerase comes and elongates it. But as it goes on to elongate, it cannot occur in that manner. For that, there is another strand, another mother strand. So for that, there is the Okazaki fragment formation, for which each time the primase has to design a primer. But as you are going on elongating, there are certain turns across the other parts of the DNA which is being loosened by the helicase. So no, it's loosened by the helicase and the topoisomerase. So helicase, it untwists the double helix, okay, and it separates the template DNA at the replication fork. The SSBPs hold the elongating strand together such that it does not fall off, and finally the topoisomerase breaks at the ends and rejoins it to get to the final template DNA strand. So these proteins work one after the other, sometimes simultaneously together, to undergo this replication. But all of these proteins are playing an important role over here. Okay. So yes. Any questions? Yeah. So that's all from my lecture part. Any questions up to here? Anyone? I'll wait for another 1 minute and see if there are any questions. Okay. So if there are no questions, I think we will go to the quiz part. Okay, where so these are the questions. I will be putting the questions one after the other, and you guys will be putting up the answers in the chat box. Okay, so yeah, let's start with one by one. So there are three questions. Let's start from question number one. Okay, so a molecule qualifies as a genetic material primarily because it must be enzymatically active, be able to store and transmit information, be present in the nucleus, be chemically complex. So all of you must try to give an answer, because these are not very complicated. Okay. Okay. So be able to store and transmit the information. Yes, that's the correct answer. Option B. Question number two says Griffith's experiment demonstrated the phenomenon of mutation, conjugation, transformation, or transduction. Okay. 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 So there is a. Okay, I have got three answers over here: A, B, C. Very interesting. So first is mutation, second is conjugation, third is transformation. So these are the three options with which most of you are quite confused. So the first, let's try to get a recap of the Griffiths experiment. What he did, he took a virulent strain, it he injected it in mice. He took a virulent strain, he injected in mice. He heated the virulent strain, injected in mice, and he took the avirulent strain and added it to a heat killed virulent strain and injected it in mice. I'm not worried about the results, what he got, about the mice being alive or dead, what happened. But you need to understand what these individual terms actually mean: mutation, conjugation, transformation, and transduction. I will not go into the depth of this. Mutation is basically when there is a particular change in the nucleotide or a segment of the DNA that is happening. Over here, virulent and avirulent strain, it's being said. There is no information that is given about uh, whether the virulent strain was, the DNA segment was removed from the virulent strain to get to the avirulent strain. So nothing, all the sorts of informations were not being discussed over here. What he said, he simply said it's being injected. So it's being injected means when you are putting a foreign genetic material into an organism, it is called transformation. Okay, so now there are different ways of putting the genetic material. So there is transduction, conjugation, but there is not two organisms. There is not like there are not two organisms that are coming in close contact before the transfer of the genetic material. So there is no conjugation that's happening. So it's simply you take a foreign thing and you're putting it in the another organism. So that's simple transformation. Okay. So Griffith's experiment demonstrates the phenomenon of transformation. That is option C. Right. The next one says which observation supported DNA as genetic material? Avery MacLeod McCarthy experiment, Hershey Chase experiment, Biddle Tatum experiment, Griffiths experiment. Okay. What I have not said over here is that there can be also multiple options that can be correct, right? So put up your answers in that manner. you can put up. If there are multiple options that can be correct, you can put it up. Right, so people have given A, B. I have not got C. So, okay. Did I get a C? Yeah, A, B, D. Okay. So, right. So, what does Avery MacLeod McCarthy experiment say? So, in simple, this experiment was this experiment basically showed that DNA is the genetic material and not proteins, right? And it was basically a transfer or a extended study on the Griffiths experiment. So he took the components of the heat killed virulent bacteria, and then moved ahead with studying the heredity and things like that. So this is also, so option A is correct because it's an extension of the Griffiths experiment. Griffiths experiment definitely tells you about the DNA is a genetic material, and Hershey-Chase experiment is also an indication of this particular theory. So A, B, and D are correct. Okay, number 4. In Hershey-Chase experiment, 32P was used to label DNA because DNA contains phosphorus, proteins lack sulfur, DNA lacks sulfur, proteins contain phosphorus. Okay. Okay, I have got A, I have got AC, AC, okay. See, you also need to understand the question. Not all questions are MSQ. So, usually, if it's an MSQ, I don't know if it's normally said or not, or just meant that it's an MSQ, things like that. But you need to understand the question first. In Hershey-Chase experiment, 32P was used to label DNA because. So only it has been said about 32P isotope. Nothing has been said about sulfur. So we will not bother anything about the sulfur as of now. So the only option that is correct over here is DNA contains phosphorus. We will not make any comments about DNA lacks sulfur, which is also correct. But we will not comment just because only 32P information is given. If there were any other option, like DNA contains phosphorus, proteins do not do not contain phosphorus, things like that, then we can comment on that. Otherwise, let's not comment about the sulfur thing. If there are information about the phosphorus which is correct or incorrect, we will only comment about that, right? Because that's the trick of the how you're answering the MCQs or the MSQs, okay? Because there might be multiple options which might seem correct, but just based on the question, it gets incorrect, okay? So otherwise, C is also correct, right? But we will stick to only option A for this particular question. The next one, that's question number 5. The glycosidic bond in nucleotides is between, quite direct, glycosidic bond is between sugar phosphate, base phosphate, sugar base, sugar sugar. Very direct question. Option C, yes. Thumbs up. Correct. Sugar and base. Okay. Right. Number 6, to the right side of your screen, a nucleotide consists of nitrogenous base, pentose sugar, phosphate group, fatty acid. another very simple question. A, B, C, yes. Nitrogenous base and pentose sugar is a nucleoside. Nitrogenous base, pentose sugar, phosphate group is a nucleotide. Very good. A, B, C. Question number 7. Which virus demonstrated RNA as a genetic material? Which virus demonstrated RNA as a genetic material? Yes, that's option C, TMV, or the tobacco mosaic virus. So it was done by Conrad Singer in 1957. So why is it? So anyone can tell me why is that only in this particular virus this fact could be proven that RNA is a has a genetic material? Yes, it does not have a DNA. So if it does not have a DNA, how does it? So does replication occur in such viruses? Does it have enzymes like the reverse transcriptase? Yeah. So it has enzymes like the reverse transcriptase because these are RNA viruses. To store the information, it cannot store the information in the form of a DNA because it does not have a DNA. But at the same time, if it has to undergo a replication, which it usually does not, I think, but if it has to send some kind of information, it has to be in the form of a DNA, and therefore there are enzymes known as the reverse transcriptase that comes over here. Yeah. Okay. Quite good. Okay. Now let's get to number 8. Which bases are pyrimidines? Adenine, cytosine, thymine, or guanine? Which bases are pyrimidines? Yes, B and C. Correct. Okay. The next one. B and C are correct for question number 8. Question number 9. Polarity of DNA strand arises due to hydrogen bonding, base pairing, phosphodiester linkage, sugar puckering. Okay, A, C. Okay, I have got 3A, 1C. Okay. Anyone else? Base pairing. Okay, I've got B. Hmm. Anyone else? Anyone else? No. So I've got A, B, and C. Okay. So the correct option over here is option C. That is the phosphodiester linkage. So polarity, what is polarity in simple terms? It's the directionality of a DNA strand. So how is it determined? It is determined by the way the sugar and the phosphate groups are linked together to form the backbone. As of now, there is no hydrogen bonding, there is no base pairing that is coming into role over here. So specifically the phosphodiester bonds connect the 3' carbon and the 5' carbon to the next sugar, and this is happening along the 5' to the 3' and on the other end the 3' to the 5', and that is why there is a directionality that is coming over here. Second, when it maintains the directionality, then comes the hydrogen bonding on the base pairing, right? So therefore phosphodiester linkages or the linking of the sugar and the phosphate groups are important for the, or play an important role in maintaining the polarity of the DNA, right? Okay. The next one, semi-conservative replication implies. So what do you understand by semi-conservative replication? One parental strand per daughter DNA, both strands newly synthesized, complementary base pairing, one strand conserved. Yes, one parental strand per daughter DNA. That is the correct answer over here. Okay, good. Now, number 11. If a DNA molecule has 20% adenine, guanine percentage will be. So, what will be the percentage of guanine if a DNA molecule has 20% adenine? Yes, B. Why? Because adenine percentage is 20%. That means thymine will also be 20% because adenine percentage equals to thymine percentage, Chargaff's rule, and guanine and cytosine are same. So 30-30 each and 20-20 for adenine and thymine. So 30% of guanine will be there. So option B. Now, the next one is, that is question number 12. Multiple origins of replication are characteristic of prokaryotes, virus, eukaryotes and plasmids. I have already discussed about it. Option C, that is eukaryotes. Correct. Question 13. DNA polymerase cannot initiate synthesis because. This is a very nice question. Why cannot DNA polymerase initiate synthesis? It lacks helicase activity, it cannot synthesize RNA, it requires a free OH group, it is unstable. Very good. It requires a 3' OH group, to which it Question 14. Proteins involved in relieving torsional stress during replication. So there is a torsional stress that is happening during replication. So which of the proteins is involved in it, in releasing the torsional stress? C, SSP protein is not correct, because SSP proteins are basically for holding this. Topoisomerase, yes, topoisomerase is involved in the doing that. So what it does, it sometimes breaks it, sometimes unwinds it, and then it relieves the torsional stress due to the formation of the bubble or during the elongation process. Right. Question 15. Okazaki fragments are synthesized in direction 3' to 5' away from the fork, 5' to 3' towards the fork, 5' to 3' away from the fork, or 3' to 5' towards the fork. So I've got a I have got a C, right. So 3' to 5' away from the fork. Nothing happens in the 3' to 5' direction in a DNA replication, because DNA polymerase cannot do it, right? So either it has to be 5' to 3' towards the fork, which is the case for a leading strand, and 5' to 3' away from the fork, that is the case for a lagging strand or the Okazaki fragments. So option C. RNA primers are removed and replaced by DNA in bacteria by. So how are RNA primers removed and replaced by DNA in bacteria? DNA pol 1, DNA pol 3, primase, DNA ligase. A, yes, DNA polymerase 1 removes the RNA primers using its 5' to 3' exonuclease activity and fills up the gap with the nucleotides over there. Yes. So A is correct. 17. Which enzymes requires ATP hydrolysis during replication? So which enzyme requires ATP hydrolysis? Yes, helicase. So helicase unwinds the DNA double helix that requires the supply for in the form of ATP. So it consumes energy. So uh, it requires energy to unwind the double helix. Now the next one is lagging strand synthesis is discontinuous due to antiparallel nature of DNA, low processivity of polymerase, weak hydrogen bonds, presence of RNA primers. A, yes, because of the antiparallel nature of the DNA and DNA polymerase not being able to synthesize in the 3' to 5' direction. The last two questions. Which feature distinguishes RNA from DNA? Presence of phosphate, sugar type, phosphodiester bonds, nitrogenous bases. I've got B as an answer. This is a very interesting question as well. Which feature distinguishes RNA and DNA? Both B and D are correct over here. Why? If it would have been only about the ribose and deoxyribose, it would have been only sugar. That's B. But over here the nitrogenous bases are there because it's speaking about the entire strands. RNA and DNA. So the nitrogenous bases vary, the sugar types vary. So B and D both are correct over here. Now the question is if. Ideally, this question should not be coming as an MCQ. It should be an MSQ. If it comes as an MCQ, what would you give? An answer. So basically, your answer should be option B. That's the sugar type, not the nitrogenous base. If it's an MCQ, if it's an MSQ, go for a B and a D. And the last one, which statements are true for DNA ligase? Joins Okazaki fragments, requires ATP or NADH, forms phosphodiester bond, synthesizes RNA primer. A, B, A, C. Okay, so I've got A, I have got B, I have got C. Good. So DNA ligase, it joins the Okazaki fragments. So what it does, it seals the nicks between the Okazaki fragments on the lagging strand of the DNA during replication. It is an enzyme that requires a source of energy, typically ATP or NADH for the catalysis of the reaction. It so as it requires ATP or NADH, so it catalyzes the formation of the phosphodiester bonds between the 3' OH of one fragment and the phosphate group 5' phosphate of the other, and it links the continuous strand, and it does not synthesize RNA primer. That is done by the primase. Okay, so A, B and C, all three are correct. Okay, right then, any questions from your part? Any questions, any doubts? I will be sharing the presentation, and this video is anyways being recorded, so I will be uploading it in YouTube as well and sharing the link with the NPTEL team who would be sharing it with you people. So right, if you have any questions, you can ask me even in the next class in this before I start for week 2. I would be so right, you can ask me in the next class in the starting, right? So no questions. Fine. So thank you for joining. We will again meet in the next week on Friday. Yep, thank you all.